Good evening, Namaskar. Welcome to HORTALK 48. Today the speaker is Dr. Tripti Vashisth. She is from University of Florida, USA. She will speak on diagnosis and nutrient management in horticultural crops. On behalf of Progressive Horticulture, I Shiv welcome the speaker Dr. Tripti Vashisth and you all to this edition of Hot Talk. Now I request Dr. Tripti Vashisth to start the talk. Madam, please.
Thank you, Shiv. Let me share my presentation. Can you see my presentation?
Yes, it is there.
Okay, all right. So, good evening, everybody. My name is Tripti Vashisth. I am an assistant professor at University of Florida. Before I tell you more about myself, thank you to the organizers for inviting me to this talk. And this is a wonderful program that you guys are doing. So I'm really happy to see this program, and I hope that I can continue being involved in such program. So about myself, so I was born and brought up in India, and I have spent pretty much all my childhood in Uttar Pradesh, in this part of the India, and I did all of my schooling in Lucknow. And if you are from Lucknow, you would know the school CMS. It's a big school there. So I've been there. And then after my schooling, I went to Bundelkhand University to get my bachelor's in technology, biotechnology. And I was there from 2002 to 2006. After my BTech in biotechnology, I moved to United States. So this is the map of US. You all probably know this. For your reference, I have marked all the big cities. And then here in US in 2007, I came to University of Georgia to do my master's in food science. So although the degree was in food science, but the topic of my research was more of a post-harvest topic. I worked on muscadine grapes, and we were looking into drying methods of muscadine grapes so that their nutraceutical value can be preserved and they can be used in food ingredients. After my food science master's, I joined University of Georgia Horticulture program for PhD, where I worked on blueberries. Blueberries are a very important crop in all over US. Actually, the production of blueberry has increased more than 10 times in last decade. So it's a very, very important crop, and I was looking into the fruit drop of blueberries. I finished my PhD in 2013, and after that, I came to University of Florida, and I joined University of Florida as an assistant professor in 2014, and I have been here since then. So about University of Florida, this is the map of the state of Florida, and here, this star here, that is where the main campus of University of Florida, Gainesville campus is. And I am at a research center called Citrus Research and Education Center, which is here, number 1 here in yellow circle. So it's about 2 hours away from the main campus. And as the name suggests, this is a research center which is focused on citrus. So just some facts about CREC. CREC was Citrus Research and Education Center, was established in 1917. So we have been here for more than 100 years. 2017 is when we celebrated our 100th anniversary. This was originally named as Citrus Experiment Station, and it was built because of a lot of efforts from the growers. So the growers wanted a research center to help with their research needs, and they actually gave the money to the state to build this experiment station. So We are very well supported from growers, and we are always working in collaboration with the growers. Now, CREC is the first and the largest research center of UF. And also it's interesting that CREC is one of the largest research center in the world dedicated to one commodity, citrus. And citrus is very important for the state of Florida and US. Before I get into the citrus production, here is the website of CREC. If you are interested in citrus or the people who work here, I recommend going on this web address, and you can find a lot of information. There are pathologists, entomologists, all different sides of agriculture. You will find people from all different sides of agriculture. So CREC is dedicated to citrus. Florida has a lot of citrus production. Actually, citrus is one of the important crops of US, and the main citrus producing regions in US are Florida, which is this, California, Texas, and Arizona. And each place has its own unique, I'll say, varieties that we produce. So Florida is basically sweet oranges, the oranges that we use for juice processing. California produces a lot of mandarins, so the fresh fruit varieties. Texas produces lots of grapefruit, and they are pioneer for grapefruit. And then Arizona produces mostly lemons and limes. This is a citrus production in United States and in Florida, and it's kind of sad to see. Actually, the citrus production is continuously decreasing in US and in Florida, and there are a number of reasons for this reduction. One, every other year. So here from 2003, 2004 to 2004 and 5, you see a big dip. And that was because we had 3 hurricanes, which affected the industry tremendously. And hurricanes were in Florida. And pretty much because Florida is the largest producer of citrus, whatever happens in Florida, that affects the US production. So the trend in Florida is pretty much what the trend you will see in US. So we had a lot of hurricanes. We had another hurricane in 2017, 2018, so another big dip. But also here we found HLB, and I'll talk more about HLB in next coming slides. But citrus production is decreasing. It's because of these problems, and also the consumer demand has been decreasing. People are getting more. They are not wanting sugary drinks, and for some reason, although citrus or orange juice has a lot of health benefits, but people are often concerned with the sugar quantity in the juice, and they are not drinking it. So there is a lot of effort in US in educating people about the nutrient benefits of citrus or orange juice, and just not to look at the sugar content of the juice. Now, talking about Florida citrus industry, I already mentioned Florida is the largest citrus producing state in US. This is the map of Florida, and all these gray colored counties are the ones that produces citrus. So pretty much most of the state produces citrus. This star is where CREC is. So we are pretty much located in the center of the whole citrus producing region. So citrus industry, it's a $9 billion industry, and it employs about 80,000 people. We are primarily sweet orange industry. In past 15 years, our production has decreased by 70%, which is quite significant. And the main reason is Huanglongbing, or citrus greening. So citrus greening was first found in 2005 in Florida. So again, this is the map showing all the regions. We found HLB in this Miami-Dade County for the first time in 2005. And here is a map from 2009. And within 4 years, so this is where the first time HLB was detected. Within 4 years, HLB was spread to the entire state. Wherever the citrus is, HLB is present. Currently, we have close to 100% infection. So every grove has infected trees. In fact, in every grove, 95 or 99% of the trees are infected. So it is a big concern for us. HLB is a bacterial disease. The bacteria has not been cultured yet. We do know that the bacteria is a phloem-limited bacteria, and in Florida, it is the candidate is Liberibacter asiaticus, and it is spread by an insect vector called Asian citrus psyllid. So HLB basically is a big concern here. And if you are in Florida and you're working on citrus, everybody is working on HLB. That's the reality of here, and so am I. I'm also working on HLB primarily right now, but overall, my responsibilities are conducting research on citrus physiology and production needs of the industry, and then developing extension and outreach program. Our growers are really important to us, and we take extension pretty seriously. So we do focus a lot. And my position specifically is 70% extension, which basically means that 70% of my effort should be on extension programs for the growers. So for research efforts, I have two main areas. One is citrus nutrition and grove management strategies, and the other one is citrus flowering and fruit physiology. To be very honest, my training has been in fruit physiology, all my undergrad, master's, and PhD. When I came here, I learned that nutrition is an important topic for the Florida citrus growers, and I started working in nutrition. So this is a clientele-based research that I'm doing, and that's how I help with my extension program. So if any one of you is interested in extension program, it's really important to know what your clientele needs, and you have to mold your research in such a way that it helps them. For my extension efforts, other than conducting research that will help growers directly, applied within few years, I am the editor of this Florida Citrus Production Guide. This is an annual guide. We publish it annually with the most updated research. It's a joint effort from entomology, pathology, and horticulture. I lead the horticulture section. I also have I write regularly lots of extension publication, organize field days, conduct different types of educational programs, workshops, and then also come up with new programs to keep growers engaged with us. It is, if you work with growers, you know, it is difficult to keep them engaged because they are looking for real information, something that they can apply soon. So coming up with extension programs is often difficult, but you have to be innovative. We started something like a citrus nutrition box last year, and I will talk more about it in follow-up slides where I'll show how the research contributed to coming up with an extension program. But overall, like I said, HLB is everywhere in Florida. So whether it's my research effort or extension effort, both are focused on HLB. And this is the center of everybody's research pretty much in CREC. Now, let's talk about HLB because I have mentioned this word so many times so far. So here is a tree. Under normal conditions, when it's growing, it photosynthesizes, produces carbohydrates, and the carbohydrates are translocated to the fruit, flowers, and the root. Now, Asian citrus psyllid, so this is the insect. It's really teeny insect. So when Asian citrus psyllid feeds on the leaf of citrus, during salivation, it transmits the bacteria. When the bacteria is in the phloem, bacteria really likes phloem. It starts thriving in the phloem, multiplying. And then after a while, within 3 months or so, we start seeing phloem plugging in the phloem system of the tree, which basically means that the carbohydrate translocation is disrupted because there are there's plugging happening. And with the disruption of translocation, now the roots don't get enough carbohydrates, so you start seeing diminishing roots, and the fruit and flower are also suffering because they're not getting enough carbohydrates. As a result, water and mineral nutrient uptake reduces, fruit quality lowers, and fruit drop occurs. Overall tree growth arrests and declines. So every aspect of the tree is getting affected. Now, there are a number of strategies that have been tried, or people are researching on those strategies to combat citrus greening. So this is the flowchart again showing insect feeds on the leaf and then what happens. So psyllid control, which will be an entomological approach that is being done. Then there are different approaches where they're trying to genetically modify psyllid so it won't transmit bacteria. They are also trying to develop cultivars which will be actually resistant to citrus greening. Either they don't have phloem plugging or they do not let the bacteria thrive in them. We have also tried some therapies like thermotherapy or antibiotics. They have not had much success, but we have tried those. Then there has been a lot of focus on improved root growth, complete and balanced plant nutrition. Also, complete and balanced plant nutrition helps with fighting the bacteria. And so this is what people have been trying, and this is complete and balanced plant nutrition and how it can help with a better plant defense response has been my area of focus. In my presentation, I will be talking about that. Now, talking about mineral nutrition in citrus, mineral nutrition and its management, diagnosis, and HLB nutrition will be the three main topics that I'll be discussing. The first two are pretty basic, so I won't spend too much time on that because it's pretty much same in all the different crops. But HLB and nutrition is something that where a lot of research is being done every day. You will see new information coming out, and there is a lot of promising information that actually mineral nutrition or fertilization has a lot to contribute to the plant. So I'll focus mostly on that. So getting started with that, first, why citrus? Of course, my job is on citrus, so I work with citrus, but also physiologically citrus is a challenging crop to work with. And the reason is it's an evergreen tree and it's a perennial fruit tree. So you always, being evergreen means that there is some amount of photosynthesis happening in the tree throughout the year. The leaves are always there. So you have to come up with a fertilization program that meets trees' requirement throughout the year. Now, there are a lot of evergreen crops of evergreen fruit crop, but citrus is a bit more challenging. And here's a picture that I like to show. This is a famous artwork. This is sweet orange Valencia, and you will see in a lot of the restaurants or places in Florida, you will see this artwork. But I find it really interesting, and the reason I would like to show it is, if you look at this one, what you see is there is a mature fruit ready to be harvested. You have flower on the tree at the same time, and you have young developing fruit at the same time. So think about the nutritional requirement or the amount of stress this tree goes through in spring. And that's what we have to work with. We have to work with three different types of plant organs on this tree. And because everything is going to affect the yield, so your nutrition program has to be such that it will support everything. So citrus becomes a little bit challenging for those reasons. And actually, just something to tell you, Valencia would be one of the few species in the world, or fruit species, where you will have the fruit is on the tree for almost 14 to 16 months. There are not many crops where you will see that. So there is a fruit on the tree always. There is no break for the tree. Now, to the basics, mineral nutrition. There are 17 elements that are essential. We know about them pretty well. Out of these 17, carbon, hydrogen, and oxygen make up about 95% of the tree biomass, but the plant are capable of taking these nutrients from their surroundings in form of carbon dioxide, in water, so plant can take those. During photosynthesis, then it produces carbohydrates, and then these carbohydrates eventually make proteins, organic compounds, and fatty acids. But to make these proteins, organic compounds, and fatty acids, that's where the mineral elements are needed. So the 14 mineral nutrients that we have to have in our fertilization program, they are needed to make these building blocks of the tree. Before we get into the elements, nutrition management is very complex, and you have to have a good understanding about it. Here are two graphs. They are from citrus crop, and they kind of show that N and K, potassium and nitrogen, which are macronutrients, it's not that you increase the fertilization and the fruit yield will increase. There is an interesting concave relationship between the two, and you have to be in the optimum range to get the best yield. And same, soluble solids or titrable acidity decreases as you have more nitrogen applied. So you have to have that good sweet spot to get optimum quality and yield. Also, good nutrition management is critical because it affects many diseases and physiological disorders. So here's a picture of apple. Bitter pit physiological disorder, which is directly related to calcium. There is no cure for this disorder. Calcium fertilization helps, but again, there is no cure. So this kind of underlines the importance of nutrition. And then also there are a number of work that you will see. This mineral nutrition and plant defense is what exactly I've been looking into and how this works. So here's a graph I've taken from one of the review articles on potassium, and you can see when you have a potassium deficiency, it affects number of pathways in the plant. And jasmonic acid, which is one of the main plant defense hormone against herbivory, and then also salicylic acid, which is involved in pathogen or infection and attacks. Those pathways are affected by if you have nutrient deficiency. This is some work that I had done on peaches, where with the nitrogen fertilization, we saw that the total antioxidant capacity decreases if you have too much nitrogen. So deficiency is not good, but too much nitrogen is not good. So again, we have to be in the sweet medium spot. And this is another paper that's very interesting. If you're working on sulfur, I suggest reading this paper. This is from Europe, and where they had found that sulfur directly affects jasmonic acid hormone and salicylic acid hormone metabolism. And jasmonic acid and salicylic acid both are phytolexins, and they are really important for the plant defense response. So if you have sulfur deficiency, you will see problems. So there's a lot of evidence that shows that mineral nutrition is more important than just the yield. Actually, it affects the plant defense response. Now getting back to the nutrition management. So fertilization is an essential part of crop production, as I explained, for multiple reasons. And there are 14 essential nutrients. They are divided into macronutrient, secondary nutrients, and micronutrients. This is one of the Liebig's law of minimum that I always like to focus on. And the reason is that according to this law, your crop production or your yield production is dependent on the on the minimum, whatever the least you have, it's dependent on that. And the reason I like this is a lot of people focus on NPK, and the micronutrients are often overlooked, and they don't apply those in the right amount, and that will affect your yield. So all nutrients are critical. So as important as it is to have all nutrients, it is also important that all the nutrients are in the right ratio. Macronutrients, as the name suggests, they are the macro, or they are in big, huge amounts in the plant, and they make up about 80% of the plant biomass. Then you have secondary nutrients and then micronutrients. So they all are important, and there is a ratio. And this is an example that I often show it to my growers. This molecule, it's chlorophyll. And here what you can see, 1 magnesium is needed for 4 nitrogen. So this ratio is really important. And in Florida, what I've been seeing that people are not focusing so much on this ratio of nutrients, and they don't see good results because of that. So ratio is really important. Now, method of fertilization is also really important because that's going to affect your efficacy of the fertilization. Soil applied, we do know soil applied, it can be in form of granular or traditional fertilizer, control release fertilizer, or fertigation. Fertigation is where the nutrients are applied with the irrigation. So fertigation. But basically in soil applied, your nutrients are in a dissolved form. And then as the water is taken up by the xylem stream, the nutrients gets taken up, and then they are distributed uniformly in the plant. Then foliar nutrients are applied in spray form. You have to have young flush on the tree in order for these new foliar uptake to happen. And because citrus has a really thick cuticle, this is a citrus leaf, and you can see it's very hydrophobic and it is a thick cuticle. So if you are applying nutrient to this leaf, there is a very limited chance that it will take any nutrients. So you have to have young flush. And also your method of fertilization should be based on whether you are trying to apply mobile versus immobile nutrients, because they both have different mobilities, or the nutrients, as the name suggests, have different mobility in the plant. Quickly, mobile and immobile nutrient. And again, one more thing, the reason why I'm talking about all of these things is it has become the basis of the HLB and nutrition research that I'll be eventually talking about. So the mobile nutrients are the nutrients that can easily move within the plant. So these mobile nutrients can go in xylem and phloem stream of the plant. They can get in and out of the leaf. They move in all directions. And basically, nitrogen put all the macronutrients, and 2 of the secondary nutrients are mobile nutrients. And the way you would know what nutrient is mobile versus immobile is if you ever see the deficiency to first show up in the older leaves, then it is a mobile nutrient, because for example, nitrogen will move to the new growth, and you will start seeing deficiency symptoms in the older leaves. So for mobile and immobile nutrient, because they can easily move within the plant, soil applied and foliar applied, both are good. It doesn't matter which one you are doing, applying. But for immobile nutrient, as the name suggests, the nutrients actually do get logged in the leaves. Wherever you apply, they stay there. So they really don't move through in the plant. Immobile nutrient will stay where it is. So depending on when you have applied your fertilizer, but generally speaking, you will start seeing deficiency in the new leaves or the new growth, because if you have not applied your fertilizer to the new growth, they will show more deficiency symptoms. Eventually, the deficiency symptoms will, if not corrected, will show up throughout the plant. But again, immobile nutrients are the ones when you will see new growth showing the deficiency symptoms. Most of the micronutrients are immobile, and for them, soil applied nutrient would be more adequate. Although one of the interesting thing is that, at least in Florida, what I've seen is most of the micronutrients are applied in a foliar form. And the reason for that is that micronutrients are needed in such a small amount that if you try to spread 2 pounds of manganese in an orchard, not every tree is going to get a granule. So foliar gives a better coverage, and people have been applying foliar micronutrients for a long time. Now we do know that micronutrients are immobile. So then in order to have good efficacy of your foliar program, you have to have a regular application, and also you have to time these application with the growth. So if you see new flush, that's when you need to apply these micronutrients, otherwise it's of no use. So all of this information goes into how your fertilization program should be. Then also, in order to have a good fertilization program, you have to understand, or you have to know what the nutrient deficiency symptoms look like. I am not going to spend too much time, but these are some of the symptoms that you will see. Like I said, micronutrients being immobile, you always see in the young flush, and then macronutrients is what you see in the older leaves. So nutrient deficiency should be corrected if you see that, but often it's said if you see a nutrient deficiency, your yield is already compromised by that time. And in Florida, at least for the micronutrients, pre-HLB, it was always a catch-up game. Growers applied micronutrients only when they saw the deficiency, and that's why their yields would have been down already by the time they applied these nutrients. Now, talking about HLB and nutrition. First, HLB. Here is a tree that shows the proper green yellowing of a branch here, and that's where the name Huang Longbing came from. So Huang Longbing is a Chinese word, or in the Mandarin, which basically means a yellow dragon. So the disease has originated from China, and as the name suggests, yellow dragon, so they would say that there is a yellow dragon on the tree. That's why you see the sectoring of yellow leaves, and you would see yellowing. But if you look closely at the leaf, what you would see is a blotchy mottle pattern on the leaf. Now, initially when HLB was found, it may have been here for a long time, but people were confusing HLB with the nutrient deficiency, because a lot of the chlorotic pattern or the corking of the vein that you see looks like a nutrient deficiency. So boron has vein corking. You will see a similar vein corking in the HLB leaves. And like zinc has islands, chlorotic islands, and you would see something similar. So it has been often confused with nutrient deficiency, and nutrient deficiency has been often confused with HLB. The way you would know whether it's HLB or it's a nutrient deficiency is, we call it a simple pen test. Okay, sorry. Okay, here's the pen test. You have a HLB leaf, and you have a leaf which shows a nutrient deficiency. So in order to know whether it's HLB or nutrient deficiency, you can draw 2 circles on either side of the middle lamella. And if it is HLB, the chlorotic pattern or the blotchy mottle that you will see will be non-uniform. So HLB is always non-uniform. Whereas a nutrient deficiency, most of the time, 99% of the time, would be very uniform on both sides. And this is how you would know whether it's HLB or nutrient deficiency. That's something that we often tell our growers to look at, and it's more reliable than anything else. So now talking about HLB and nutrition. Since HLB arrived in Florida, which was 2005, there has been number of reports on nutrition and HLB. Of course, like I said, the nutrient deficiencies are often confused with HLB, so a lot of growers thought to correct HLB with the nutrient applications. But these are some of the titles from Citrus Industry magazine, which is a very common industry here, common magazine here, and number of articles. I'll say every issue has at least one article on nutrition. So there is a lot of interest in nutrition and HLB, and one of the reason is, sorry, I think my slides are a little bit slow. My slide is not moving. I'm sorry for this technical thing.
Again, what you do, ma'am, again share it and again you can begin that slide, same slide.
So stop sharing and then start?
Yeah, you can stop sharing.
Okay. Is it up now?
Yeah, it is there. You can move now. You can move that slide, ma'am.
Okay. Thank you.
Yeah.
So one of the reasons why there has been a lot of interest in nutrition work is because one of the symptoms of HLB is diminished root growth. So this is a picture taken of healthy tree, symptomless tree, and a tree that has HLB symptoms all over it. And what you can see is that there is a lot of difference in the root density. So a tree that shows symptoms has almost 70 to 80% root loss. And root growth is not so much affected. It's mostly the root density. So there is a lot of new growth happening, but root density is affected. So because of this, a lot of growers started working, or they got involved in nutrition work because they thought, okay, our roots are not functional. So they tried to correct it with the nutrient deficiency. Nutrient application. Sorry, it's stuck again. My slides are stuck again. Shiv, it has stopped again. I'm going to disconnect and connect again. Will that work? If I connect?
Yeah, it will work. You can.
I'm sorry, everybody. I'm gonna try to connect again.
Audience, please send your queries and questions to progressivehorticulture@gmail.com.
Okay, I think the slides are back again. Um, Sorry about the technical difficulty. Okay, so growers in Florida, they initiated the whole nutrition movement actually, and growers started to treat the trees, whatever deficiency they were seeing in the tree, they would apply it foliarly. This Boyd, if you know anything about Florida industry, you would know Maury Boyd was one of the growers who started this whole foliar sprays. And there was a lot of work done on the nutrition. Unfortunately, most of the work resulted in inconsequential results, or the findings were not consistent. So there was this famous article with the inconsequential effects of nutritional treatment. So there has been a lot of controversy on the nutrition, whether it works or not. And there are number of reasons why, and the reasons. Efficacy of nutrient application really depends on the method of application. So in this case, most of the growers were applying foliar nutrition. I will show in upcoming slides why that might have been a reason foliar had limited success. Soil type and pH. So even if you are applying soil applied nutrient, you really have to pay attention to what is your soil type, what is your irrigation pattern, and when are you applying in the year. So nutrition is really complex. Now, some of the recent findings, this is a recent article from 2019 from a graduate student here, and what they found that high rates of manganese actually improved the tree growth and the yield. So this is the root system of a HLB tree, and this is the root system of a tree that has been treated with manganese. They call it overdose because they were applying almost 3 to 4 times more than the recommended rate, and they saw improvement. Now the key here is soil applied. They were applying manganese in the control release form, and I believe that has one of the benefits here. Like I said, manganese is an immobile nutrient, so it is really important that we apply soil applied because that's the best way the tree can take it up from the soil. Now, this is some of the ongoing work that I have been doing. It's a field trial, and then this field trial has resulted into 2 small studies that I'll show you eventually. So I have this big field trial because that's what extension involves. Big demonstration plots, something that growers can see and assess. So we have been doing this study for almost 5 years now. It's done at 2 locations. These are 10-acre plots that we are working with Valencia trees. And then we are applying fertilizer by hand in specifically the wetted zone of the tree. So in Florida, microjet sprinklers is what we use. So we exactly know in what area you should expect the roots to be. And this is a microjet sprinkler, and we are trying to place the fertilizer in the wetted zone because that's where the roots are, that's where the water will be applied. So it makes logical sense to apply specifically here, the fertilizer specifically here. In this experiment, I have been trying 10 treatments, but so there are different rates of micronutrients that I'm applying. So manganese increased by 20%. These are based on the recommendations for a Florida citrus. But this is an interesting product that I'm trying, Tiger Micronutrient. Tiger Micronutrient this is the picture of this nutrient, fertilizer. It is a sulfur bentonite clay. So this clay, with the humidity and temperature, actually it releases sulfur, as well as it has all the nutrients. So whatever your mix is, in this case it's a blend of iron, boron, manganese, and zinc. So each prill has all of these nutrients, sulfur and the 4 micronutrients. So when it starts releasing nutrients, it's releasing everything. And when you are applying it in a broadcast application or by hand, however you are applying, you have a uniform distribution of these nutrients throughout the orchard. So I really like this fertilizer because it can reduce your soil pH and it can apply nutrients uniformly. So in this experiment, I have my control treatment, which is a regular fertilizer with foliar micronutrients, and then we have different combinations. This is some of the information like how much nutrients I'm applying, um, and everything in pounds an acre, because that's what our growers use. So I'm sorry, it's not in hectares. But so the interesting thing is, so these are the two locations where this trial is being done. And what we are seeing is manganese and boron, when increased by 50% of the recommended rate, we are seeing improvement in the tree density. So the tree is looking fuller and better. And as far as the yield is concerned, so this is a 3-year cumulative yield. The first bar in both the graphs is the control. And then we have our different treatments. There is a lot of variability, but what we are seeing is that manganese treatments and boron treatments are doing good. So we are seeing yield improvement with high rate of micronutrients, CRF, and soil pH reduction. One of the interesting things, though, is again, these are the two sites where this trial is being done, and this is the total yield for the 3 years. And I have ranked these treatments based on the yield. And what you will find interesting is that control is last out of the 10 treatments in both sites. But somewhat of a funny thing is that in this site, Arcadia site, my best treatment is iron 20% increased. And that is actually my second from the bottom for the other site. And for this side, this treatment is manganese plus boron is the best treatment, and it is second from the bottom at the other side. So it's somewhat of ironical that I have good results, but they are not same at the both sides. But overall, what we are seeing is high rates of micronutrients are beneficial for HLB affected trees, and soil pH reduction. So tiger micronutrients do seem to be beneficial in this case. So however, you can reduce your soil pH. But the nutrition program needs to be customized for each site, and no one size fits all. So that's one of the message that we have been trying to give to our growers, that they really need to customize the program, their fertilizer program. Out of this research trial, I have been doing quite a bit of my extension outreach box, and we came up with a citrus nutrition box. So this is just to give an idea, some type of extension work that I've been doing. So we came up with the nutrition box. This is the box. It has a kit for leaf sampling and soil sampling. And what we are asking our growers to do is to send us leaf and soil samples. We have a lab that is doing the analysis, nutrient analysis for them. We receive the results from the lab, and then we UF faculty, so it's myself and some of the extension agents that we have. We meet, we discuss these results, and we are trying to customize a nutrition program for each grower with each sampling that we receive. So it is a tedious process, but the goal is that if we can show growers how to read your leaf nutrient analysis results and how to customize your fertilizer program based on that, they can be independent eventually. So when next time I tell them that nutrition program should be customized, they will be independent and they can do it by themselves. Right now, a lot of them rely on the fertilizer sales reps. And nothing wrong with that, but sometime, because if you are a fertilizer sales rep, you may have more interest in selling your fertilizer than actually improving the tree health. So we are trying to educate our growers about this. So this is a nutrition program that we have been doing for 1 year now, and we have a lot of success with it, and it is going to be continued. And then so that's something that we have been doing. Now, based on this research, we came up with 2 smaller studies, the greenhouse studies, and the idea was, so this work has been published, and actually this year, and this is my PhD student, Faisal, who published this work. And nutrient, this is the title, but the main goals that we were trying to see in this one was investigate qualitative and quantitative differences in nutrient uptake of HLB trees. So, like I said, higher rates of micronutrients are helping the trees. The question is, why are they helping? Exactly what is happening there? Then also we wanted to look at the efficiency of nutrient uptake in HLB affected trees, because we do know that they have smaller root mass. So are they efficient in nutrient uptake? And then we also did a transcriptomic analysis to study the nutrient uptake response. So this study was done in a greenhouse. We used a hydroponic system for these trees, this study. So this is a picture of the bucket inside. You can see we had complete 100% hydroponic system. We used aerators to keep it aerated, the water, and then we had built this trellises to keep the trees upright. We used mid-sweet orange grafted on Kaharski rootstock, and before starting the study, we deprived the plants from any fertilization for 6 months, and we did that so that the plants will develop a deficit of nutrients. When we apply Hoagland solution in the hydroponic system, it would readily take up the nutrient, and we can start seeing the differences between the HLB and healthy roots, how they are taking up the nutrient. So we did a 2-month study. We sampled every 7 days. We looked at the nutrients in the solution, nutrients in the leaf. Talking about the results, so here's a picture of HLB and healthy trees. You can see healthy trees are green, looking good, leaves are really good. And then we have our HLB trees with somewhat of a diminished root system. You see deficiencies of nutrients, chlorotic patterns, and blotchy mottle. When we talk about the fresh weight, as expected, HLB trees had significantly lower root mass, shoot mass, and the leaves. And we found that the root to shoot ratio was significantly lower in HLB plants as compared to healthy plants, which basically means HLB plants have fewer roots to support the shoot system. This is the chlorophyll content in the leaf. Here, yellow color is for the HLB plant and the green color is for the healthy plant. The dotted line is for the plants that were not fertilized throughout the experiment. And then the solid are the ones that were deprived for nutrients for 6 months, but then we applied the nutrients. So they received the nutrient. And what we do see that healthy plant and HLB plant start further apart from each other. But after 2 months of nutrient application, they both have the same chlorophyll content. So with fertilization, the chlorophyll content of the leaf can be improved, which basically means you can improve hopefully you can improve the photosynthesis rate in the HLB leaf. So fertilization is promising, no doubts. Now looking at the leaf nutrient analysis. So these all panels are showing you the leaf nutrient concentration, and in each panel you will see what nutrient it is. Here you have healthy plants which were fertilized for 2 months, HLB plant which were fertilized for 2 months, and then we have healthy and HLB plants which were not at all fertilized throughout the study. And this is where we find some interesting results. What we see, the ones in the red square or the blue square are the ones where we saw HLB plants showing some sort of deficiencies. Now, healthy plant were also not fertilized. So let's talk about calcium here, for example. This black bar with white dots in it, that's the healthy plant not fertilized. Healthy plant not fertilized. This plant, even though was not fertilized, but the calcium level in the leaf was same as the healthy plant that was fertilized. But when you look at the HLB plant, when you don't fertilize it, there is more deficit of nutrient. And that's the case even in potassium, sulfur, manganese, iron, zinc, and magnesium. which basically is telling us that whatsoever the reason is, HLB plants are metabolizing or using these nutrients or losing these nutrients at a higher rate than the healthy plant. So if you have a HLB plant, you have to focus on these nutrients. Healthy plants were more forgiving, so you can fertilize them less and they'll be fine. But with HLB, you have to focus on these nutrients. And the interesting thing was also that the secondary nutrient, which is calcium, magnesium, and sulfur, even after fertilization, they were never as had the same level as the healthy plant. So this is really interesting. There's something going on with the magnesium, sulfur, and calcium. Like I said, sulfur is involved in plant defense response, so is calcium and magnesium, and it was pretty interesting. Now, when we look at the nutrient uptake efficiency of the plants, so this is the nutrient uptake efficiency graphs. We have HLB plants and healthy plants, and this is milligram per kilogram of root mass. So we are comparing 1 gram of root from HLB plant to 1 gram of healthy plant root. And what we found that HLB plants actually have higher nutrient uptake efficiency. So if 1 gram of HLB root compared to 1 gram of healthy root, HLB root are working harder or they are more efficient in nutrient uptake. So this is really neat to know that HLB roots are working harder to take up the nutrient. And possibly it is because they have the diminished root system. So because they have a smaller root system, so now the fewer root have to work harder to take up those nutrients and meet the tree's need. We also looked at the vascular bundle. So this is the root system, anatomical sections of the root system. And here the first two graphs, or these two are the day 0 plants, HLB plant and healthy plant. We saw that HLB plant have a smaller diameter of the vascular bundles in the root system. But upon fertilizer application, we saw a significant increase in the vascular bundle diameter. And that was we saw in both in healthy and HLB, but in HLB it was more significant. The increase was more significant. We are looking now into if this increase in the vascular bundle diameter contributes to a higher nutrient uptake, because we do know that they have higher nutrient uptake. We also did a transcriptomic analysis, and transcriptomic analysis, we did RNA-seq analysis. So RNA-seq is used for a global gene expression study. We did it on day 0, 7, and 14, and we did a qPCR analysis as well to confirm the RNA-seq results. Quite interestingly, what we found that when on day 0, HLB and healthy plants are compared, there were only 9 differentially expressed genes. So if you are not fertilizing a healthy plant and HLB plant, they're pretty much similar in response. But when when you apply fertilization, we start seeing more differentially expressed genes. So there is a change in response upon fertilization. These are the 9 genes that were differentially expressed on day 0. So these are really important genes. I'll not go into each gene, but we do see a zinc transporter being upregulated to 6-fold. There's another zinc-related one, and there were a lot of stress-related genes. Upon fertilization, we saw 30 genes. So we saw a lot of transport-related genes being expressed after fertilization in HLB plants. So blue bar means these genes were upregulated in HLB plants. We saw quite a few transport and nutrient-related genes, out of which 30 genes were directly related to nutrient uptake. And we also saw ZIP10 being significantly upregulated in HLB affected trees. So there is an upregulation, which means, again, that healthy tree, when we compare HLB trees to the healthy trees, HLB trees are have an upregulation. So they are spending, or they are trying hard for the uptake of these nutrients. This is a figure that is showing the pathogen pest attack pathways and different pathways involved. And again, upon nutrient availability, several plant biotic and abiotic response pathways were altered in HLB plants. So when there was no fertilizer, we did not see this happening. But when we applied the fertilizer, we see abiotic stress response. We see quite a few PR proteins which are involved in which are pathogenesis related to genes, signaling, and hormones. So all of this is happening only when the fertilizer is applied. So to conclude from this study, we find that HLB affected roots are functional and efficient in nutrient uptake. HLB affected trees metabolize nutrients at higher rate than healthy trees. To improve nutrient uptake efficiency, the roots are going in some anatomical and transcriptomic changes. And HLB affected trees should be supplied with constant nutrient and a higher rate of micronutrients. So they do actually need higher rate of nutrients. And nutrient availability allows the trees to respond to biotic and abiotic stresses. So this study is pretty important. Now, going to the next study, because in the field trial, we did see that with the reduction in soil pH, the trees were performing better. So we have recently published this work too. My master's student published this work. Here, we were looking at the effect of irrigation water pH on performance of healthy and HLB root. And the two questions that we were trying to answer were to investigate the effect of irrigation water pH on performance of healthy and HLB root and the molecular regulation. So why we are looking into irrigation and pH, irrigation water pH? We do see that trees yield and health improves with reduced soil pH. There is other researchers have also shown that HLB symptoms intensify at high soil pH, and the root density is inversely related to high soil pH. Now in Florida, the optimum soil pH is 5.5 to 6.5. It has been historically. But our irrigation water, because we are sitting on a lime aquifer, so we have a lot of bicarbonates in our water, and the pH is pretty high. And because we irrigate with this water, our soil pH has been over 6.5. Now, but it has been 6.5 for decades. Pre-HLB, we were not doing any soil acidification. So the question is, why now soil acidification has become more important? So what is HLB doing that we have this? Now, it could be that HLB is biotic stress, and then high pH is abiotic stress, which affects the water and nutrient uptake, and therefore a poor performance. We also know that soil pH is going to affect the nutrient uptake. So we did this experiment in a greenhouse. We again used mid-sweet on Kaharski. And we irrigated the plants with pH water at pH 5.8, 7, or 8. And we had healthy and HLB plants. We used the sand collected from a grove in order to have somewhat of a real world situation in the greenhouse as much as we can. So we used Candler fine sand, which was at pH 7.2. On day 0, we fertilized the plants, and then we started irrigating the plants every 3rd day with whatever pH they were supposed to receive. This study was conducted for 60 days. The original plan was to conduct it for 120 days, but because the plants started to die in our extreme treatment, we had to cut short it to 60 days. At 60 days, we collected the tree health data, and RNA-seq analysis of the roots was performed on day 60 samples. Now, results. So here, in most of the graphs, upcoming graphs, what you will see, x-axis is the 3 pH treatment. Then the pattern or the textured bar is for HLB, and the solid bar is for healthy plants. So what we did see that at pH 8, we saw a significant number of plants died within 60 days. Um, so at pH 5.8, no HLB plants died. At 7, 12.5% of the plants died. And at 8, 37% of the plants died. This was significantly higher from the healthy plants. Then we looked at the change in height of the plant. And again, what we saw that at pH 8, there was a very less amount of increase in the plant height. Now, the disparity between the healthy and HLV plant was interesting, because even though healthy plant was also being affected and had less change in height, but it was more, the disparity was more there. Whereas at 5.8, they all look same, whether it's a healthy or HLV plant. Looking at the leaf chlorophyll, we saw a decrease in the leaf chlorophyll as the pH increased, and it was same for HLB and healthy plants. We also looked at the leaf number, and what we saw that, so here the solid bar, that is again for the 5.8 healthy, and so they all start about the same, but because of the pH treatment, there is a more sharp decline in the leaf number in pH 8 HLV plant as compared to the 5.8. So this is 5.8 healthy, this is 5.8 HLV and 5.8 HLV. So we saw a correlation. This is a correlation plot, and we did see that for the HLB plant, we have a R square of 0.99, whereas for healthy, it's not as much as HLB plant. So there is a relationship between the leaf drop and HLB. We also see a decrease in the leaf biomass as expected. As the pH increased, the leaf biomass decreased. Here is a picture of the plant. Bottom one is the HLB plant and the top one is the healthy plant. At 5.8 pH, actually, we saw a lot of new growth happening in HLB plants, more than healthy plant. But at pH 8, we saw pretty much the plants were dying. They were dead. And the 7 was intermediate. When we look at the root biomass, we did not see a difference with the pH, but we saw that healthy plants have more root biomass than HLB. But when we looked at the root system, the differences were more apparent. Again, the bottom one is HLB and the top one is healthy. And if you look at the 5.8 pH, HLB plants are showing a lot of new growth happening. But at pH 8, there are barely any roots there. So there was an effect of pH on the root density. We also looked at the EC of the soil, which is basically electrical conductivity. So if the tree was taking up more nutrients, you will see a lower EC value. And we did see that 5.8 had the lowest EC among 5.8. Healthy had a lower EC, and pH 8 had highest EC. So the nutrient uptake was affected at high pH, and it was more affected in HLB plants. And we did see that the soil pH it was rebounding. So even though we were watering the plants with 5.8, 7, or 8, there was a rebound in the pH. So the plants were trying to stay closer to the 7, which is the natural pH of the soil, 7.2. Now we did look at the leaf nutrient analysis. We could not do a statistical analysis on that because most of the leaves had dropped. So we had to pool the leaf samples. But we did see a very significant decrease in the zinc, magnesium, and calcium of the leaves. So according to the optimum range, there was quite a bit of decrease in these nutrients, and specifically in pH 8 HLB plants. So whatever the reason is, somehow these trees, pH 8 pH 8 HLB plants were getting more affected. Then, as I mentioned, we had done RNA-seq analysis on these trees. So we looked at the total number of genes, and what we found that, so here is pH 5.8 healthy versus HLB, pH 8 healthy versus HLB. And we found that 520 genes were upregulated in HLV plants and about 700 were downregulated. But at pH 8, about 270 genes were upregulated in HLV plants and nothing was downregulated. When we looked at this comparison closer, so now we are looking at pH 5.8 HLB versus healthy plants, and we did a GO analysis. The major pathways that we saw were at 5.8, HLB plants had more response to bacterium, immunoresponse, response to oxidative stress. They were detoxifying these reactive oxygen species. There was more secondary metabolite production happening, and all of this secondary metabolite or reactive oxygen species are known for plant defense response. They are involved in plant defense response or response to bacteria. So we were seeing that the plants were showing more pathways that are related to plant defense response, and these genes were upregulated. Again, this is where the root growth was more. And then we also saw a lot of root system development genes, shoot system, so a lot of development, and possibly these genes contributed to the growth that we saw. When we look at the down-regulated genes in HLB plants compared to healthy plants at pH 5.8, we saw jasmonic acid to be down-regulated, and we saw senescence to be down-regulated. And we do know that when the plant is under stress, it produces more jasmonic acid, or there is more senescence going on in those plants. So this was interesting that if these down-regulated genes are all positive regulators of jasmonic acid, we are seeing a reduction in the stress in the HLV plants at pH 5.8. When we looked at the pH 8 HLV versus healthy plants, so these genes are the ones that were upregulated in HLV plants, we saw actually upregulated genes, more root development genes. We saw a lot of programmed cell death happening. So these are the ones that show a lot of death of the root, and there's a lot of programmed cell death. There's a abscisic acid response, and there's some immune response. So there's cell death, immune response, and then we see growth and development happening in pH 8 HLV plants. So altogether, what we see in this study is that high soil pH, higher death rate in HLV plants, high soil pH, higher leaf drop in HLV plant. Visual symptoms show that more reduction in root growth. At low pH, though, the growth is comparable between healthy and HLV plant. At high pH, there is a reduced chlorophyll content, and at high pH, there is less leaf biomass. Now, looking at the conclusion from the molecular analysis, at low pH, we see higher growth, more development, and more ROS detoxification. At low pH, we see downregulation of stress, and at high pH, we are seeing more programmed cell death and senescence. So altogether, this study tells us that there is an interaction between HLB and soil pH. HLB plants showed higher growth at low pH. Healthy plants are not significantly benefited at low pH, but HLB plants are. And the effect of high pH to plant extends farther than just nutrient restriction. So yes, at high pH, the nutrient uptake is limited, but we are seeing a lot of molecular regulation of the the negative contributors to the tree health. So there is more than nutrient uptake with pH. So altogether, from my presentation, I'll wrap up my presentation with the take-home message that we are seeing that nutrition can improve plant defense response. and HLB affected trees do benefit from good nutrition program. Micronutrients are critical for HLB affected plants and are needed at slightly elevated level. Due to small root system in HLB affected trees, nutrient uptake is limited, but HLB trees are efficient in nutrient uptake. The fertilizer program needs to be site specific, and soil pH affects the growth of HLB affected trees. And nutrient uptake contributes to the tree growth, but in HLB condition, soil pH is also contributing to the tree growth, and the relationship is beyond the uptake of nutrients. So all of my conclusions are related to HLB, but that's what I have been doing, and that's the focus of Florida Citrus is currently. With that, if there are any questions or any discussion, I'll be happy to take the questions. Also, here is my email address. If you have any questions, you want to send it to me later on, please do feel free to do that. Thank you.
Thank you, madam, for wonderful talk. Audience, please send your query and question to progressivehorticulture@gmail.com. And the response of audience was tremendous with very encouraging remarks. We are highly grateful, madam, to you for sharing time and delivering such an important talk. Thank you, audience, for your active participation and support.
Okay.
And
thank you.
We have one question. If you want to reply, we can show on the screen. We have one question. Anup Srivastava asked what the percentage of citrus growers are covered under the regular leaf analysis program in Florida, and how much time do you take to complete the leaf analysis once the grower deposits the leaf samples to your laboratory?
Okay, so I think Dr. Srivastava, you asked specifically for the nutrition box program that we are doing. So this is more of a demonstration to the growers. We have limited number of resources, as you would expect. So this service is provided to each grower, any grower in Florida who is interested in participating, but they get maximum of 3 boxes, which means 3 nutrient analysis 4 times a year for 1 year. So as far as the groves are concerned, for each grower, we have about 30 acres covered. That's how much we can cover. Right now, we have about 100 growers participating, or 100 boxes. So I'll say about 100 acres is what we have covered so far. The goal is to educate them, and then eventually they can start using their leaf nutrient analysis by themselves. So right now, the goal is to educate. We are using a lab to do the nutrient analysis for us. So they do the nutrient analysis, we get the results, and then we make recommendations for each grower. We meet every week to discuss any results that we receive. It does take some time, but I don't know how to answer how much time it takes, but it takes time. It's definitely a time-intensive process.
Thank you, ma'am. Thank you a lot, ma'am.
Okay.
Thank you, ma'am.
All right. Thank you. Have a good evening, everybody.
Yes. Good night, everyone.